I'm going to talk about asbestos and its properties that make it a carcinogen. I find that fascinating, even though it's tragic on an individual level. I'm going to start first by talking about the morphology, the shape of the asbestos that makes it a carcinogen. I'm going to talk about what asbestos does within the lung when it makes it there. Then finally, a 3D look at asbestos. So size matters. Here's a look at the lung. And if you look at the lung, you will see that, of course, you've got the trachea coming down, and then it splits into two, and that splits into two, this splits into two. And after 23 divisions, we get down to the alveoli, which are the little air sacs that are at the very bottom, and that's where the air exchange takes place. The good air goes in, the bad air comes out, the oxygen goes in, the carbon dioxide goes out. And there are 300 million alveoli, and they're just barely big enough that you can see them with the naked eye. The surface area inside the lungs, this is pretty incredible, even though those alveoli are so small. If you were to spread them out, they would fill half a volleyball court. That's huge. That's like 4 times greater than the surface area of your skin. So a very effective way of passing gases back and forth. Now, large particles are removed. When the large particles come down at very high velocity, they have sufficient momentum or inertia that they impact at these bifurcations or at the forks. So you get all these large particles collected here, and that's good news because you've got the mucociliary escalator. The mucociliary escalator has goblet cells that discharge the mucus, and then it's got the ciliated cells that sweep this mucus in one direction. So you've got the air passing back and forth here through the tube, and then the mucus is swept to the pharynx. Goes up to the pharynx and you go, and it's gone. That's good news, good riddance. So all the big stuff gets deposited in the upper lungs and gets carried away by the mucociliary escalator, but the small stuff ends up in the alveoli. So particles that are smaller than about 2.5 microns get past there and can be deposited in the deep lungs. So 2.5 microns is actually 1/10,000th of an inch. That's a very tiny particle that makes it down to the alveoli. Now here's what's interesting about asbestos, is that you can have a fiber that's 5 microns long, and if it's only 0.55 microns wide, it has an aerodynamic diameter of 2.5 microns. Therefore, it makes it into the lung. If you have a fiber that's 50 microns long and is only 0.46 microns in diameter, it's going to make it to the alveoli. Can you see where I'm going with this? If you've got a fiber that's 1,000 microns long and is only 0.45 microns in diameter, it's going to make it to the lungs. So the lungs were not made to handle these very long particles, and I'll tell you why in a second. When they get into the lungs, they're minerals, they're rocks. They're going to be there for a long time because they won't dissolve with the typical bodily fluids. We look here at this picture, we see that this is an alveolus right here, one of the air sacs, and you can see an amosite fiber right here inside the air sac, and then back here, white blood cells or macrophages, and I'll talk about those now. The macrophages, the white blood cells, are mobilized. They come in and they carry the fibers to the lymph system. And these are mostly short fibers because the white blood cells are about 5 microns in diameter. They're big enough to be engulfed. Is that good news? Well, not necessarily, because the recent studies by Suzuki and Dodson, Ron Dodson, who is on the ADAO scientific board, have shown that the mesothelial tissues that they've looked at are loaded with short asbestos fibers. So short is not necessarily safe. And the rest of the fibers, they're in your lungs for life. Here's a picture now of the white blood cells, or the macrophages, and asbestos fibers. And you can almost imagine this as a storming of the castle. The outer defenses, which is a mucociliary escalator, have been breached. And so all these very thin but long asbestos fibers have made it into the air sacs. And so it becomes hand-to-hand combat with these white blood cells. So it's very easy to imagine that this white blood cell here is saying, all right, you're coming with me to this little short fiber right here, throws the cuffs on that fiber and carries it away. But the fibers like this are just going to laugh and say, ha, you can't do anything with me, and it remains there. So CDC, on their website, have talked about the carcinogenicity of asbestos. They say that asbestos is a known human carcinogen. That's no news to any of us here. And they also say that all types of asbestos are carcinogenic, but some scientists believe that the amphiboles may be more potent in causing mesothelioma than chrysotile. Some scientists, so there's some controversy there. However, they continue, both types can cause mesotheliomas and are believed to be equally potent in causing lung cancer. So what are some of the hypothetical mechanisms for toxicity? Fibers generate the free radicals that damage cells and DNA. The white blood cells are always secreting lysosomes that they use to dissolve foreign bodies like bacteria, but it doesn't work on asbestos because asbestos is a mineral. So you've got all these free radicals running around that are going to cause damage to cells. They stimulate proliferation of target cells. That is, they secrete growth factors, and these growth factors can enhance growth of the epithelial or lung cells. And uncontrolled growth, of course, is cancer. They can act as delivery sites for chemical carcinogens, and the prime one that I think of is benzoylpyrene in cigarette smoke. So you've got a fiber sticking outside the alveolus like this, and a benzoylpyrene molecule comes in, and it adheres to the asbestos fiber because of the surface property of the asbestos fiber. So now you've got this benzoylpyrene molecule that is available to get into the bodily fluids and cause cancer. Finally, this one's a little bit easier to understand. Long asbestos fibers have been shown to interfere with the mitotic spindle. That is, they interfere with cell division. And 15 years ago, workers in our laboratory published a paper in Cancer Research showing this very phenomenon. If you look here, you see the nuclear envelope of a lung cell. And here are the chromosomes. I'm sure you've all seen time-lapse videos of cells dividing in the mitosis and the chromosomes pulling apart like this. Well, right here we've got the crocidolite outside the nuclear envelope. And if you look by 34 minutes, some of the crocidolite is starting to penetrate inside the nuclear envelope. And you get down here and you see that there's quite a bit of crocidolite inside the nuclear envelope. And when they get to that point, you can see that right here, this crocidolite fiber is touching this chromosome right here, and DNA breaks off. A little bit of the chromosome breaks off right here. And then in every subsequent cell division, the daughters are lacking that DNA. So when you get DNA missing or perturbed like that, it can cause mutations, and mutations, of course, can lead to cancer. So we're going to look at fibrous talc. When I say fringe asbestos, these are not your typical 6 regulated asbestos. In fibrous talc from New York State, it's a commercial variety. It's not stuff you put on your baby's bottom, but it's used for industrial applications. And it is a mixture of talc and fibrous amphiboles. And fibrous amphiboles, like anthophyllite and tremolite, are asbestos. The other one is going to be the Libby amphibole asbestos, which does not fit into the classical definition of asbestos because it's mostly winchite and richterite. These were all taken in our laboratory with our scanning electron microscope. And so there's fibrous talc from Balmain, New York. And that's about, 2 millimeters across. But that stuff is incredible. It's like dried, super dry firewood. You just touch it with your fingers and it starts to break apart. And if you look down here, you can see things are just kind of dangling in space. You look a little closer, and you can see these very small fibers here. And these are the ones that are going to make it into the alveoli. Here's a look at it from the end. Another picture of the fibrous talc. You see some very serpentine fibers there. And then this is an amphibole bundle from Libby vermiculite. And this stuff looks like little grains of rice in the vermiculite that's in, 35 million attics right now in the US. And it looks fairly innocuous because a grain of rice ain't going to make it into my lungs, no way. But what I've done here is I've taken a needle and I scratched a little furrow right down here in this bundle. And you look at the furrow of the needle, and what do you see coming out here? The unfriendly fibers. And so this is what it looks like when you start to break apart one of these little bundles of amphiboles. And here's another bundle, and if you look inside here, we'll take a close look at that. Sean, that doesn't look like a cleavage fragment to me, does it?
No, sir. No,
okay.
Sir.
Then finally, Tony Ward at the University of Montana, sent me some tree bark samples several years ago and said, take a look at this stuff. It was from trees near the mine in Libby, Montana. And I thought, there's not going to be anything there. But I looked, and you can see a fiber here. And if we look a little more closely, you can see this, pay attention to this little oval right here. At low magnification, you can hardly see any fibers. This is about the view that you'd be seeing with a light microscope. But then you get the electron microscope and go up in magnification. You see again this little oval shape here. Now you're starting to see some fibers. Go up higher in magnification. There are even smaller fibers. And you go up higher in magnification, even more small fibers. So this is what's on the tree bark out near the Libby mine. And the people in Libby, of course, use the firewood for burning. So they're investigating what the possible impacts of the contamination of the tree bark is in Libby. Finally, this is a project of a grad student of mine several years ago. She cultured human lung cells on some of the Libby amphiboles. And you can see the lung cells here and the Libby amphiboles.